Hello, everyone. Good evening. I'm Diana Pullman, Executive Director of PANDAS Network. Just a reminder to any parents watching, we also have a video available to you from last month's webinar. It's on our YouTube channel. Tonight's event was sponsored with the wonderful support of PANDAS Physicians Network. Thank you to the many donors who made this event possible. Tonight's webinar will review the latest trends in translational research in PANDAS and PANS children and how these important developments have moved basic science research to direct evidence of the impact of this illness on children's brains. Please welcome Dr. Jitan Agayu, neuroscientist, Columbia University, and chair of the PANDAS Network Scientific Advisory Board. And I just want to say on a personal note, Dr. Agayu has been a friend to our children for over a decade and a wonderful colleague who supported many of the physicians and parents in this new and developing field. I will now turn you over to our esteemed host, Susan Boas, Executive Director of PANDAS Physicians Network and the President of the IOCDF Board of Directors. I'm signing off. Thank you both so much for this wonderful event.
Thank you, Diana. It's such a pleasure to be partnering with you today.
Thank you.
As Diana said, I'm Susan Boas. I'm the Executive Director of the PANDAS Physician Network. We're a nonprofit that provides PANS and PANDAS resources to clinicians while supporting research initiatives. Our vision is that PANS-PANDAS will become easily diagnosed and treated with accessible therapies that dramatically reduce suffering and lead to a cure for those patients. We are so excited to partner with the PANDAS Network for today's webinar. It is entitled Looking at the Brains of PANDAS-PANS Children, How Peripheral Infection Triggers Brain Inflammation. We will also have the opportunity to discuss the latest research from the Agalu lab, including the recent paper, Distinct Th17 Effector Cytokines Differentially Promote Microglial and Blood-Brain Barrier Inflammatory Responses During Post-Infectious Encephalitis. But before we begin, a few reminders to the attendees. Today's format will include a presentation followed by a Q&A. The Q&A will include both questions submitted during registration and also during the webinar. There is actually a chat function where you can submit questions to our presenter. Please remember that questions should be research-oriented and relevant to the presentation topics. We cannot provide medical advice. Finally, a recording of this presentation will also be made available in approximately 2 weeks. So with those housekeeping notes taken care of, I am delighted to welcome our presenter, Dr. Jitan Agayu. Dr. Agayu is a neuroscientist with over 20 years of research experience in developmental, cellular, and molecular neuroscience. He is an associate professor of pathology, cell biology, and neurology at Columbia University's Irving Medical Center. He has met with us before to discuss his research, and we are very eager to hear what has transpired since we last spoke. I am very excited to turn this over to you and look forward to learning from you this evening. Jitan, you'll need to unmute yourself. There you go. Okay, thank you, sir.
So thank you, Susan, for that wonderful introduction, as well as Ina, and I want to thank both PANDAS Physician Network and PANDAS Network for organizing this event. So we had one event organized by PANDAS Network in the middle of April 15, where I basically discussed some of our recent research, mostly tailored toward parents. Today's discussion will be tailored predominantly for the physician and physician care healthcare providers. So it will be a little bit more scientific and medical research based. Okay, good evening, everyone. So what I would like to do tonight is tell you our recent studies to really understand this big question of how peripheral infections, in particular, peripheral infections triggered by group A strep infections, trigger brain inflammation in particularly PANDAS and PANS. So before I start, a few disclaimers. A lot of this work was funded by an NIH grant from the National Institute of Mental Health and some support from other funding sources from the National Institute of Health. Some of the work that I'll be describing with the human sera was initially funded by a small grant and from the International OCD Foundation and the Camper Basic Research Grant from Columbia. And of course, some of the work was funded by very generous donations from PANDAS Network, Newport Equities, and many small donations from a lot of parents and advocates and supporters. So if you're online, I want to thank you for your support of this research. So of course, the pathogen that I'm going to focus most of the discussion today is really group A strep infection, because group A strep is a gram-positive bacteria that is commonly the agent that causes the sore throat in children, but it has also been associated in a variety of autoimmune sequelae, targeting the heart, causing rheumatic fever, the kidney glomerulonephritis, and then in the brain, the predominant neurological sequela of rheumatic fever has been described as the Siemens chorea, and that recently in the 1990s, Susan Sweeto and colleagues, studying Siemens chorea, they really kind of, identified and described for the first time PANDAS, which stands for Pediatric Autoimmune Neuropsychiatric Disorders Associated with Group Strep. And what really understand about the mechanisms of autoimmunity related to group A strep is this really concept of molecular mimicry that Madeline Cunningham and colleagues have put forward, in that basically the, when you develop an immune response toward group A strep infection, that immune response then aberrantly recognizes proteins in the surface of the various target organs, and these antibodies then target these organs, triggering an autoimmune response. And so what we do know is that Siemens chorea is very common after severe group A strep infections. The main region of the brain that is targeted is the basal ganglia. And then several work initially from Madeline Cunningham and also more recently from Chris Pittenger at Yale University have in fact identified that there are a lot of circulating antibodies, either against dopamine receptors or more recently, like in PANDAS from cholinergic interneurons that target the basal ganglia and drive some of the motor symptoms. And then in addition to that, at least in the context of dopamine 2 receptors, work by Madeline Cunningham has really elucidated that these antibodies recognize and bind the dopamine 2 receptor, and they trigger the activation of the neurons. But PET imaging studies have also shown that indeed in this region, there is activation of the microglia. And for a long time, it has been known, even though very much less characterized, that there are neuropsychiatric symptoms in patients with Siemens chorea. And then, of course, in the 1990s, the pediatric autoimmune neuropsychiatric disorders associated with strep were described by Susan Sweeto. This is also caused by repeated bouts of group A strep infection, again, targets the basal ganglia. And the predominant, abnormalities are psychiatric, particularly a sudden onset of OCD. But, some of the motor systems are also largely affected. Studies by Chugani and colleagues have characterized the presence of inflammation in this region. However, as we know, and has been a challenge for a lot of clinicians, is that this disease is really difficult to diagnose. And there are very few or very poorly characterized biomarkers that can be used. And so one of the some of the fundamental questions that we've been interested in terms of understanding about these syndromes is the question of how do these peripheral antibodies that are generated against group A strep and then may target the brain reach the brain. The second is if there is, there's been described, there is like activation of microglia, but one of the main questions that, we had is what mechanisms drive and sustain this persistent microglial inflammation in the syndrome. And then potentially, by understanding the disease, can we potentially develop novel tools to for either the diagnostics or potential treatment. And also, there's a very ill-understood mechanisms, whether this is really an autoimmune or autoinflammatory disease, and I'll touch upon at a little bit at the end of the talk, as well as in the Q&A session. So really the central question in my laboratory has been really trying to understand what are some of the mechanisms that drive both neurovascular dysfunction and neuroinflammation in these syndromes, which I'm calling them post-infectious encephalitic syndromes. So of course, this is a very difficult question to address in humans because we don't have in samples, and really we only have mostly blood and to a certain extent CSF. But this is sort of a very good question to address in animal models for disease. And animal models have been fundamental to us to understand mechanisms of disease in many neurological disorders, including stroke, multiple sclerosis, Parkinson's disease, Alzheimer's. And it's important to note that not every animal model recapitulates every aspect of the disease. So the model that we decided to use in our studies was a model that will mirror the naturalistic infections that you get with that children get with group A strep infection, where in this case, in mice, we would take young mice that were just weaned from their mother. So this would be equivalent to young children. And then we'd perform intranasal infections weekly with live group A strep infections for several weeks to mirror the repeated infections that children get as they grow. And then at the end, after a month or so, we would harvest the brain tissue for analysis. And pioneering work done by Patrick Cleary at the University of Minnesota and colleagues has really shown before that these repeated intranasal infections drive the generation of T cells and adaptive immune response in the nose. And in particular, one region of the nose that in mice is equivalent to the human tonsils and adenoids is a tissue called the nose-associated lymphoid tissue. And you can see in this image here that the group A strep is labeled in green, and you see a large number of T cells surrounding this bacteria in the nose. It's important to note that group A strep does not have an affinity for the brain. In fact, you never find group A strep infections in the brain. So we're not really talking about an infection of the brain per se. It's an infection of the nose, which is quite far away from the brain. However, one thing that drew our attention in the initial studies is that we know that indeed in the nose, there are neurons that are responsible for the sense of that convey the sense of smell to the brain. These are these cells here in green, and they're termed olfactory neurons. And so what is interesting about these neurons is that they have little processes called dendrite on the surface that are important for sensing smells. They have a large number of expression of olfactory receptors, but their axons project into the brain. So we postulated that perhaps this might be a route by which the cells that are generated in the nose to fight group A strep infection there might travel into the brain. And in the initial study that we published in 2016 in Journal of Clinical Investigation, in collaboration with Patrick Cleary's group, we very nicely demonstrated just that in fact, what you can see here is an image of a naive mouse and a mouse after repeated group A strep infections. And what is very prominent to see is that indeed these T cells labeled here with a marker CD4 are crawling along the olfactory nerve and entering the brain, whereas you can see they're almost completely absent in the naive brains. And when we look at the number of these T cells in the brain, they're highly upregulated after 5 infections, but basically there's barely anything after 1 infection or in the naive mice that have never received repeated infections, intranasal infections with group A strep. And so the question that we had is what pathology do we get in the brain after intranasal group A strep infections? And because the study is published, I'm just going to run quickly through it. But the presence of the T cells in the brain is associated first with a breakdown of the blood-brain barrier. This is a structure here that where endothelial cells that line the vessels of the brain form this very tight barrier that prevents the free diffusion of molecules, ions, antibodies from the blood into the brain. And it's important to preserve the function of the brain. But in the context of this, when you have T cells, the cell-cell junctions are disrupted, as well as there is an increased transport of the antibodies from the blood into the brain, which can be visualized in this particular image. In addition, what we did find is that when you have T cells in the brain, there is a state of microglial activation. In this case, what we're calling activation, and I'll go more in depth into, is really expression of a lysosomal marker called CD68, which can label also the infiltrating macrophages. But you can see here that after intranasal group A strep infections, there is a massive upregulation of CD68 in microglia, which makes them more phagocytic. The final thing I want to point out is that indeed one of the consequences of this is that expression of excitatory synaptic proteins, such as VGLU2, is completely downregulated in the after following infections with group A strep, and that leads basically to a loss of neuronal activity. So when these mice are exposed to odors, they don't seem to smell, and then there is basically like very little firing of these neurons when they are presented to an odor. So the question that we had that we wanted to address is from our histopathological studies, we knew of an array of, we had just touched the surface. We knew that there are changes, histopathological changes occurring at the level of multiple cell types in the brain. But we wanted to understand this a little bit more in depth. And a very talented former graduate student in the laboratory, Charlotte Wayne, she decided to take unbiased approach to do is using some of the state-of-the-art technology. And so basically in the state-of-the-art technology, you can dissociate the brains into single cell. We can we can either perform single cell RNA sequencing of all the cells, or we can enrich for basically specific cell types, endothelial cells or immune cells to analyze. And the single cell RNA sequencing gives you like a much more in-depth information about how cells are changing the RNA levels, and so what we call transcriptional states of the cells. So what was very interesting and important, so what you're seeing here is a representation of this data from a lot of experiments, but you're seeing here, these are called Tisney plots, and what you can see is different populations of cells in the brain, neurons, astrocytes, microglia, endothelial cells, and so on and so forth. In the bottom, you can see that these are separated now by state, where the color, the black color is the healthy, and the blue is basically after group A strep infection. And you can appreciate that a large number of cells like macrophages, neutrophils, T cells, they're all blue, suggesting that basically the cells are normally not there in the brain, and all of a sudden they're infiltrating the brain after repeated group A strep infections. But there are cells that are there, but they're certainly changing their state. And the two cells that mostly change their state, one was endothelial cells. You can see this shift from a black to blue. And then the second population is this large population of microglia that also shifts their state during disease. So what are these? So we were very intrigued about endothelial cells and microglial cells because we had seen that these cells are changing very dramatically in the context of disease. And so we analyzed these cells in much more depth. So what you're seeing here is just isolating the endothelial cells and performing that analysis. You can see that the endothelial cells separate into two distinct states, the healthy state on my right and the diseased state on the left. And you can see that there are a large number of genes that are upregulated in disease and a large number of genes that are downregulated. Important to note that indeed the antigen presentation and the endothelial response to LPS are highly upregulated, but like things like blood-brain barrier genes are downregulated. And this is an example of 3 unique blood-brain barrier genes. The names are not very important, but you can see that basically in these violin plots where we can map the expression of these genes, their expression is significantly downregulated after group A strep infections. This is from a computational standpoint. This is looking at the expression of that RNA in tissue. You can see that this gene ITM2A, which is a very BBB specific gene, is highly downregulated after group A strep infection. And so this transcriptomic data really supported our initial functional studies that indeed blood-brain barrier function is highly compromised when the T cells are entering the brain after repeated group A strep infections. So the next cell type that we focused were microglial cells, because of course there have been a lot of studies in children, some studies in children by Chugani and colleagues stating that indeed there is microglial activation in the brains of children with PANDAS and PANS. And these cells have been associated with a large number that they play a critical role in a lot of neurological and psychiatric disorders. And so what we did is that basically this is what you're looking here is again a Disney plot of all the microglial cells. Each dot is a single cell isolated from either the control or the brains of control mice or those with group A strep infection. And you can see that we have 6 distinct clusters. 2 clusters we call homeostatic. These clusters are predominantly present in the healthy brains of mice. And then 4 clusters are disease-associated clusters, or we call them streptococcal response microglial clusters. And when you see here the expression of homeostatic genes, it's highly enriched in clusters 1 and 2, and the disease-associated genes are basically clusters 3, 1 of the disease states. Now, particularly what was interesting is that a lot of these genes that we call disease-associated genes are the same genes that get upregulated in microglial cells, also in a large number of neurological diseases like experimental autoimmune encephalitis, a mouse model of MS, or Alzheimer's disease. And so they're kind of hallmarks of disease state. But when we look a little bit more in detail, then and we look at what genes are differentially expressed from the control state to the disease state, 3 classes stand out. One of these classes is basically antigen presentation, which is a set of genes that is important for basically presenting, peptides to T cells and activating T cells. And in fact, microglia is one of those cells that we call an antigen presenting cell. Is that basically its role in the brain, it's one of the innate immune cells of the brain whose role is to trigger the activation of T cells in the brain. So you can see that a lot of these genes that are important for activating T cells are really upregulated in the context of disease. The second one is the response to type 1 interferon. And this is a little bit, unclear because, this is typically is a pathway that is triggered when you get a viral infection. But a lot of recent studies have seen that this is really a classical way of how, microglial cells get activated in this state. And the third class that really is highly upregulated by the cells is the production of the cytokines and chemokines. These are proteins that are secreted by one cell and basically allow other cell types. It's a mechanism to bring other immune cells to the site where there is an infection or there is inflammation. And so it's a mechanism to basically communicate, by which immune cells communicate with one another. However, these proteins are not, neutral to the other cells. Endothelial cells, neurons respond to these proteins, and they change a lot of their function, as I'll describe a little bit later. And this is sort of what we call a heat map, where you can see the expression of some of these genes. So on top here, we have the antigen-presenting genes, highly upregulated after group A strep infections. So the yellow colors indicate like high expression. And then some of these chemokines, and I'll come back to some of these, but in particular CCL2, 3, 4, and 5, and CXCL10 are very low in the microglia in the brain, in the mice, in the healthy state, and they get highly upregulated in the context of disease, as well as these two pro-inflammatory cytokines, IL-1 beta and TNF-alpha, and then of course a lot of the interferon response genes. So the next question that we had is we wanted to understand, this disease-associated microglia. Where or how are they distributed in the brain? Are they uniformly present throughout the brain, or are they in distinct regions of the brain? And to address this question, you can't address this question with a single-cell RNA sequencing, but we took an approach called a spatial omics approach, where we use this technique called multiplex error-robust fluorescent in situ hybridization, or MERFISH, to spatially visualize all the cell gene expression changes in this region of the brain, in the olfactory bulb. And so when we use this technique, and I'm not going to go a lot in detail, but they're described in more detail in the bioRxiv paper that we have deposited, what became very interesting is that indeed, when you look at the cells that express a lot of these disease-associated genes, they're predominantly present in the outer side of the olfactory bulbs, whereas basically the homeostatic genes are scattered uniformly, particularly in the healthy state throughout the brain. So that suggested that indeed there is not a uniform spatial distribution of this disease-associated microglia in the brain. So what triggers this ununiform expression? And when we look at basically the distribution of the T cells are not always uniformly expressed. So when we look at the structures, the T cells are predominantly present in these glomerular layers. This is the layers when the nerve cells from the periphery come in and make connections to the brain. And this is the hub, the first hub when the T cells come in. And so when we look at the distances of the T cells with the microglia, the closer the distance is, the more that microglia will have disease-associated states, as illustrated here. So when you're mapping here the distance of the microglia to the nearest T cell, you can see that basically that distance is very small in the outer side, the glomerular layer, where we have a lot of T cells, and that basically it's much longer in the granular region, where you have much less basically disease-associated cells. So really what this data is telling is that that neuroinflammatory response is very localized. Basically, T cells have to be in close proximity to microglia in order to trigger disease-associated states in microglia. And so this really changes our perception that there is this uniform big inflammation of the whole brain. That's not true. We really see a very regional, local cell-cell communication happening in the brain. So far what I've told you is that I've told you that indeed during disease, what we have found through the beautiful work of Scarlet is that indeed the microglial cells change their state after repeated group A strep infections. They upregulate the antigen presentation, the response to type 1 interferon, and the production of cytokines. And endothelial cells also respond by basically downregulating expression of a lot of genes that are important for the maintenance and function of the blood-brain barrier. And so in a previous study that we published in 2020 in the PNAS, a former grad student, Marianne Platt, now postdoc at Yale, very nicely demonstrated that all this pathology in the brain really is dependent on the presence of Th17 lymphocyte. This is a subpopulation of T cells that is critical, has been important in a large number of autoimmune diseases. And basically, when you get rid of these cells, you basically can rescue a lot of this pathology. And so we confirmed that with single cell RNA sequencing here. What you can see here is in gray is the healthy, in blue is the group A strep infection wild type mice, and in green is the group A strep infection ROR gamma deficient mice. These mice don't have Th17 cells. And what you're seeing here is that in the context when you don't have Th17 cells, basically those homeostatic good genes go up, the disease-associated genes go down, and the expression of cytokines, chemokines, or interferon response go down. However, one thing that keeps really going up is antigen presentation, and I will come back to that later. But to summarize this, really what happens if you don't have Th17 cells in the brain, you rescue basically the expression of homeostatic genes. You can reduce or rescue this abnormal upregulation of chemokine and cytokine, and also you can rescue the response to interferon. So this was all really important, but then we really wanted to understand something more. Now, Th17 cells can make a lot of inflammatory cytokines, but one of the ones that is really critical for their function is this cytokine termed IL-17A. And we were excited to understand if we were to target IL-17A with an antibody that binds to it and prevents its binding to the receptor, are we able to phenocopy all the phenotypes that we see when we ablate the RR gamma, when we ablate Th17 cell lymphocytes? And this is important because this might be an important therapeutical tool, as I will discuss a little bit later. And so this is the experiment that we did here, is that we used the neutralizing antibody against IL-17, and we delivered it twice a week because of the short life of the antibody, but then right before and during the course of the infections. And you can see that basically that treatment does not really necessarily prevent the generation of the Th17 cells, which are important for fighting infection, or basically their entry into the brains. However, one of the things that we did notice is that when you don't have IL-17, you can rescue a lot of the blood-brain barrier leakage. So what you're seeing here is, just like we saw before, when you have when you do the injection with an isotype control antibody, you have a massive leakage of the BBB, measured here by the leakage of serum IgG. But in the context of like IL-17 blocking, you can rescue partially that phenotype. And when we took the microglia and performed single-cell RNA sequencing, what was very clear is that, in fact, we saw a very nice phenocopy of what we'd see in the RR gamma deficient mice, in that there was an upregulation of the homeostatic genes, these are the good genes, a downregulation of the disease-associated genes, sort of a downregulation of chemokines and cytokines, and the response to interferon. However, similar to what we saw with the RR gamma deficient mice, there was an upregulation in antigen presentation. So basically, as far as we can tell from these, there is pretty much a very big phenocopy of the basically the RR gamma T mutant mice that don't have Th17 cells and IL-17 blockade. And I don't have time to talk about this, but it is in the paper. Scarlet went even further. I mean, she basically also knocked out, eliminated another cytokine called GM-CSF, which is made by Th17 lymphocyte. And she showed that that doesn't always phenocopy the same phenotypes we see in RR gamma. suggesting that indeed the blockade of IL-17 seems to be a critical parameter to basically rescue a lot of the pathology we see in mice. But mice are not human, right? And so, you know, as some of my colleagues here in the Department of Neurology say, you know, you might as well be in the Department of Zoology if you only study mice. So the question is, what are the relevance for the human disease, right? So is there anything that we found in the mice bearing or important for the human disease? And so to address this question, Tyler Cutforth, when he was in the laboratory as an associate research scientist, collaborated with Susan Swedo at the NIMH, as well as the Wendy Vargas and Shannon Lanny, who are both here at in the Department of Neurology and Psychiatry. They both have left Columbia since then, to basically collect a cohort of PANDAS and PANS cases and healthy control and perform the cytokine study in the sera. So this is a description of our cohort. This is basically the mean age of the healthy and cases. They're roughly the same. And this is the distribution by sex and the source. So this is a table illustrating the, so we performed a multiplex bead assay with 45 different cytokine, chemokine, and growth factors to identify in an unbiased way what cytokines, chemokines were elevated in the sera of PANDAS and PANS cases at the acute phase of the disease. And what we were able to demonstrate, as you can see here, several cytokines and chemokines were highly upregulated in the sera of PANDAS and PANS cases, but not in the healthy controls. And some of them were really intriguing because, and I have lighted here in red, because these were the very cytokines that we saw upregulated in microglia and macrophages in our mouse model, suggesting that indeed there is a really commonality, or there are common themes between the disease model in mice and the human disease. And when we look a little bit more in depth how like the distribution of the sera is in patients versus healthy controls, you can see that indeed there is a massive upregulation of CCL2, 3, 4, 5, CXCL10, this factor called GM-CSF, which is made by Th17 lymphocyte, and IL-6 and IL-7. So really, this data suggests that indeed there are multiple cytokines and chemokines that are highly upregulated in PANDAS and PANS cases. And then some of them could potentially be derived from either the myeloid population, either the microglia or the macrophages that are circulating, although of course there could be other cell types making this. So the question then we have, so what we have so far is that indeed, you know, in the sera of humans, there's a large number of chemokines, cytokines, and growth factors. Some of them could be derived from microglia and macrophages, and these could be potential diagnostic markers that we could use to either monitor the disease state or its progression. So the question is then, you know, do they have the sera? Do they have an effect on the human blood-brain barrier? And so in collaboration with a very good friend of mine and colleague, I think she's in the call here, Irina Peterson at the Scintillon Institute, we've embarked on developing a 3D model of the human blood-brain barrier. And in this model in particular, what we can do is we can basically culture human brain endothelial cells in the top well here in red, and then we can culture in the bottom other cell types of the brain, such as astrocytes and pericytes. And the movie here shows basically these endothelial cells in the brain that form a nice tubule. And so by moving this platform, we can basically like mimic blood flow here, and we can expose these cells to either media or sera from healthy donors or patients. So doing this study, actually, so basically here in a beautiful image provided by Dr. Irene Peterson at the Sintelon Institute, you can see that endothelial cells form very nicely tight junctions. And you can see that these other cells, perivascular cells, pericyte, interact well with them. And astrocytes can come to the point of the basement membrane to form contacts with endothelial cells, similar to what you have in the human brain. And so when we expose then this human neurovascular unit, we call it, or the blood-brain barrier in a dish, to sera from either healthy donors or PANDAS patients. And then after 48 hours, we actually put a tracer to measure the permeability. You can see that basically in the sera that were exposed to the neurovascular unit that was exposed to PANDAS sera, you can see a massive leakage of that tracer in the what we call the brain compartment or the brain inside, suggesting that this barrier becomes a lot leakier basically to tracers. And so this is actually a very strong proof that indeed the circulatory cytokines and chemokines that we identified before in the blood or in the sera from children at the acute phase of the disease open the blood-brain barrier. And so basically our findings then suggest that I would state that indeed based on our findings, we have a pretty strong evidence right now to suggest that PANDAS and PANS to a certain extent is a neuroinflammatory disease. I would be hesitant to call it an autoimmune because we really haven't been able to identify a particular specific brain target. But based on the work from Madeline Cunningham and Chris Pittenger, that they've identified autoantibodies that circulate that could potentially target the brain, there might be a possibility that it's both. It's an autoimmune and autoinflammatory disease. I do feel that many of these inflammatory cytokines, or at least the combination, is quite unique to PANDAS and PANS. I've looked at a lot of cases with COVID. I've looked at the cases of direct viral encephalitis, and the profile of the chemokines and cytokines is very different. Yes, some of them get all upregulated, like CCL2 is always high, but a combinatorial combination is really unique. And so I do think that indeed this combinatorial code could be used to basically identify the patients for the diagnostic tool and to monitor their treatment. And then obviously, given that a lot of these inflammatory markers are present in the sera, obviously pharmaceutical drugs that target the neuroinflammatory response may be beneficial in the treatment. But I think the study that we did in mice to look at the effect that IL-17 blockade has on disease outcome also suggests something even more. That in fact, some of the drugs that already exist and are FDA approved to treat other autoimmune diseases like secukinumab from Novartis or ustekinumab from Janssen, that are used really to basically treat these other diseases, ankylosing spondylitis, psoriasis, including treatment of psoriasis in children, the psoriatic arthritis and Crohn's disease and ulcerative colitis could potentially be beneficial. Now, of course, one of the major counterindications is an active infection. So I think I would necessarily, recommend using these drugs in the acute phase of the disease. But once that infection is cleared, if there is a recurrence of the disease, this potentially could be considered as a treatment strategy in very severe patients. And with that, I want to conclude and thank some of the people who have really contributed extensively to this work. Of course, the initial work was started by a very talented postdoc, Erica Smith, together with a talented associate research scientist, Tyler Cutforth, pictured here. Some of the initial work on the deletion of Th17 lymphocytes was done by Marianne Platt, featured here. She was a very talented graduate student. And some of the current work on the single cell sequencing and treatment with IL-17 blocking antibodies done by the super talented Charlotte Wayne. featured here and here. She just left Columbia to start a postdoc at Rockefeller University. So she's on for greener pastures. And of course, currently the work is being carried out by Ugur Akchan, a postdoc scientist who is really interested in looking at the genetic risk factors for the disease. Of course, some of the work would not be done without collaborators. Vilas Menon, a computational biologist, really has provided a lot of insights in this. Dori Schaefer at UMass, who has really been very helpful with the analysis of microglia and some of the Murphy studies. And of course, I am highlighting here also the funding. And some of the recent work we're doing in the human blood-brain barrier is through a collaborative consortium with Irina Peterson at the Scintillon Institute, Andrew Sproul here at Columbia, and Raphael Lis through some funding from the NHLBI. And with that, I can take questions. Thank you very much for your attention.
Thank you. That was really wonderful. And I'm always impressed by the number of brilliant minds that have come together to really help families, children that are suffering from this disease and really on the edge of science, of learning new things that are going to help, I think, a lot of families in the future. So thank you for your work and thank you for all of your colleagues. We are a very impressive list of collaborators. With that, we're going to turn to our Q&A. If you have questions and you're here on the call, you can put those in the chat or in the Q&A section, and we will try to get to them as time allows. I'm going to start with a couple of questions that came to us in advance of the webinar, and I think you addressed one already, but I'd like to ask it to you again, just to elaborate, because it's one of the most common questions we get about whether, based on this work, PANDAS and PANS is therefore an inflammatory disorder or an autoimmune disease.
Yeah, so thank you, Susan. So yes, I elaborated on this, but I want to discuss a little bit more. And so technically, I think based on our study, I think that we can definitely state is an inflammatory disease because a large number of these inflammatory molecules, cytokines, are very highly upregulated in the serum. We have some CSF and we're doing those studies, and certainly, and based on the studies that we've done with single-cell RNA sequencing in the mouse, we can clearly see that the microglia are highly inflammatory. So at least it's a very clear that there is a state of inflammation, peripheral inflammation and brain inflammation. Whether this is triggered by an autoimmune, I think it's a little bit more complex. Certainly in mice, it's more difficult to ask that question, but at least in humans, I think it's also really tricky because yes, there are antibodies against dopamine receptors and there are antibodies against cholinergic neurons that would point out that perhaps there might be an autoimmune response. Mm-hmm. But and basically there is also beautiful work in Australia suggesting that there are T cells that are targeting some of these dopamine receptors. But I think that will require a little bit more careful and thorough and further study to really nail down that this is indeed an autoimmune disease. But certainly there are a lot of autoimmune components that I think our work has identified. I mean, clearly the fact that these 17 cells play a critical role, and these are important cells in virtually every single autoimmune disease that we know, points toward that direction. I'm just trying to be a little bit more cautious in the statement, but I would say, yeah, the jury is still open. I think it still could be an autoimmune disease, but certainly I think a lot of the data point clearly to an autoinflammatory and neuroinflammatory disease.
Yeah, great, thank you. I thought two of the most exciting parts as you were presenting were the question of whether that might lead to biomarkers in combination, and then also that might then lead us to some additional medications for treatment. it feels like we've been so early in the studies that we haven't gone to new medicines. And so that was really an exciting part to learn a little bit about. We had a question prior to the webinar. Do you have putative biomarkers that may aid diagnosis and or responsiveness to treatment in PANDAS and PANS?
Of course, so I have to put a little bit my salesman hat and say that indeed, I think that several of the cytokines and chemokines that we have identified could potentially be very powerful diagnostic tools. I would say not one of them, but I think a combination of several of them. And in talking to some of the clinicians, I do know that Quest Diagnostics has already in place, assays to test several of these inflammatory cytokines and chemokines. And I know a lot of rheumatologists are using those already for, diagnostics of other autoimmune diseases. So I think that they potentially could be, a very valid and important diagnostic tool that could be used either for disease to aid in the diagnosis of the disease, but I think more importantly to monitor the progress of the disease.
I hope you'll dig deeper into that, and we'll be talking more about diagnostics.
We will, and I think one of the interesting part as we move forward is to really kind of understand whether, in fact, this can be used to monitor disease progression. Like if we were to determine the cytokine profile in children who were in remission, or after treatment with like IVIG, plasmapheresis, or other immunomodulatory, what happens to these cytokines? And I think in broader term, so we haven't really seen a difference between PANDAS and PANS. So what we have defined as PANS is the cases that we've collected at Columbia, where we couldn't identify clearly a group A strep infection in the throat, but there was a history. But of course, as you know, like a PANS can also be caused by other agents such as Lyme. And so it'll be interesting to see like to what extent some of these are similar and what is unique and what is common. And I think that is, of course, work of many, many years to come, I think, hopefully.
That should keep you busy for our generation.
Hopefully, yes.
All right, the next question plays into that. How do elevated lab values of various cytokines and interleukins play into treatment decisions and diagnoses for PANDAS and PANS? And I think they mean as of today.
Yeah, so can you repeat the question one more time?
How do elevated lab values of various cytokines and interleukins play into treatment decisions or diagnoses for PANDAS and PANS?
Yeah, so I would say I think it's very critical to really determine what specific cytokines are present, because if, for example, in our cases, we did see elevation of IL-23 in the PANDAS patients and GM-CSF. These are two of the cytokines that are highly upregulated by Th17 lymphocytes, but also a lot of the cytokines that are present or chemokines that are, upregulated by immune cells like microglia, macrophages were also upregulated. And so I think it's at the end, I think there could be a combination of different, tools, but I do think that, whether which therapy you start will depend a little bit on what specific biomarkers you see in the bloodstream. I certainly would think that if you see, an elevated number of these pro-inflammatory cytokines and chemokines that are made mostly by innate immune cells, I would say that maybe steroids would be a good therapy.
Well, that leads us right to our next question.
But I'm not a clinician, so I don't want people to take this at the face value. So I want to make the disclosure right now.
I will admit that I was just thinking about folks going to their pediatrician asking for testing of cytokine levels. And I was like, okay, we probably have some work left to do.
So cytokines you can actually test through Quest Diagnostic. That's not a problem. I think when it comes to
that's
treatment,
so helpful to tell people. I think
that becomes a little bit more complex. Yeah.
All right, so the next question comes right after that. Is the conceptual framework of the innate versus adaptive immune system wrong? Can the priming and memory adaptation of the innate immune system explain the mechanism of PANS as it can be triggered by a variety of infections?
I don't think there is, I still feel that both arms of the immunity play an important role. I do think that the adaptive immune response is critical to actually trigger, to fight infection. And also it's critical also in terms of triggering a lot of the, unfortunately, some of the pathology we see in the brain. However, we do also see a very important role of the innate immune response in the process as well, and particularly also microglia. And in fact, we have some work now to really target or address some of the mechanistic insight is what would happen in the microglia. Because what I didn't discuss is what we have seen is that IL-17A by itself cannot really break down the blood-brain barrier, the human blood-brain barrier. And so we think that there is a relay mechanism where T cells talk to microglia, and then microglia can basically trigger a variety of the histopathology because they are able to secrete a large number of these cytokines and chemokines. And so the story is has become a little bit more complex the more we dig down. And I think that in my view right now, I would say that both cells, both adaptive and the innate immune responses, that play an intricate role in the process.
We have a question about IgG trafficking related to pro-inflammatory microglial activation that drives CCL2 production, which promotes monocyte blood-brain barrier trafficking. How is this related to increased IgG trafficking across the blood-brain barrier?
Yes, so I don't think it's only CCL2 that does that. So in some in vitro studies we have done in the laboratory, we've seen that several of these chemokines and cytokines that are made by microglia as well as macrophages, including CCL2, 3, 5, they're very good at promoting the transport across the blood-brain barrier. One of the mechanisms is a caveolus-mediated transport for the aficionados and the audience, but basically, I don't think CCL2 is the only one that does that. I think there are several of them that play an important role. But I think that this also comes to the point that indeed some of the more deleterious chemokines and cytokines that affect mostly the barrier are coming from microglia and macrophages and not really T cells, but they're triggered by the presence of the T cells.
All right, great, thank you. All right, I have a number of questions in the chat. We'll try to get to as many as possible here today. I see one that asks, can this be measured? And I believe they mean the disease itself. Can this be measured in our patients in a way that supports or confirms or excludes or decreases the likelihood of PANS via currently available studies? You mentioned Quest diagnoses for cytokines and chemokines. And the second part of their question was, does this work confirm or explain the value of antibiotics, NSAID, steroids, IVIG?
So I think certainly I can start with the second one because it's a little bit faster to respond. I think it confirms several of that three-legged stool that has been postulated as a keystone in the treatment for PANDAS and PANS, because you do need the antibiotics to fight an infection, because if that infection persists, what we have seen is that you need multiple infections to trigger generation of T cells that enter the brain, and that can lead to a variety of pathological consequences. So definitely, I think antibiotics are probably like the first line of defense if you have an active infection. I think certainly the presence of this neuroinflammatory and inflammatory state, I think would warrant the treatment with potentially steroids as well. But you know, you have to be careful there. And certainly, I think IVIG and plasmapheresis is important because you want to eliminate the pathological antibodies. And you know, by diluting out this inflammatory cytokines, you're also buying time. But in my view, both the IgG and plasmapheresis are just tools to buy time. They're not long-lasting treatments. So I think that you know, therapies that would target the adaptive immune response, T cells, B cells, in my view, would be therapies that would provide a more long-lasting effect.
That is I think that's a really important statement, because most of the field has really been looking at some of those shorter term treatments as the primary treatments for a long time.
I agree. And I think, you know, we still don't understand truly how IVIG works. I mean, part of it is to dilute, of course, whatever pathological antibodies you have. So there's evidence that it can also interfere with B cells function, and B cells can communicate with T cells. But I think really there are a ton of drugs right now that target T cells and B cells. And I think for patients who are very resistant to a lot of these treatments, I think it's time to think about this more severe therapies, but they could be potentially, you know, mind-changing, similar to what has happened in MS.
Okay, thank you. That's very helpful. All right, I'm going to go to another question. This is in regards to group B strep. The touted protein critical to cross-reactive reactions by GAS is M protein, but additional mechanisms are also considered to be involved, since other infectious agents void of M proteins, such as Mycoplasma and influenza A, are documented to produce PANDAS. What are your thoughts about other strep species, i.e. group B strep, carrying non-M proteins, which can serve a similar role as M protein?
Yes, so group B is complicated because group B can infect the brain. And so any pathogen that can infect the brain can trigger other mechanisms from what I have discussed. And I think this is also important to keep in mind with Mycoplasma as well. Mycoplasma can infect the brain itself. And so in addition to like some of the, and there, once you have a brain infection, you have, you can trigger a very different set of mechanisms to what I have described. Now, to come back to that point, also staph can trigger a Th17 response, Staphylococcus aureus. And I think, you know, once you've primed the generation of T cells with group A strep, other bacteria or viruses that can also trigger, you know, can stimulate the Th17 cells may potentially exacerbate some of the outcomes. But, you know, it's a really tricky question to answer, because a lot of some of these pathogens have very specific mechanisms.
While we're talking in that realm, I don't know that you've done work on this, but I'll ask you the question and feel free to say not studied yet. Can IL-17A be activated in a PANDAS child because they're fighting fungi or mold exposure?
I mean, potentially, but you know, I think that we haven't done any work, but you know, the activation, I mean, there definitely is a possibility. And that's my point here, is that whenever you have an active infection with another pathogen that can trigger a Th17 adaptive immune response, that if you have a primed mechanism by group A strep to trigger the entry to the brain, I mean, it's potentially, it can lead to other consequences, right? Because then you're breaching a little bit the blood-brain barrier, you're priming those T cells to traffic more to the brain. And so once those T cells begin to traffic more in the brain, there is a potential that they can recognize antigens and they can, then trigger an immune response. That could also be said for antibodies, right? I mean, one of the things that is sort of not discussed very well is even healthy patients have a lot of antibodies that target the brain, but they're not sick. And so, potentially, if you have these other forms of infection that may prime the blood-brain barrier to be open, then some of these antibodies can enter the brain.
We actually have a few questions very similar to that, so thank you for answering that. So a couple of folks have asked questions about the phrase repeated streptococcal infection. So we have two questions. I'll give you the first. Please discuss whether or not timing in between recurrent streptococcal infections or timely treatment of strep impacts likelihood of developing PANDAS.
Yes, so I think that this is an important lesson. We don't know about PANDAS and PANS, but I think we certainly know more on this in the context of MS, right? MS is a relapsing, initially presents as a relapsing remitting, that then eventually can develop into a secondary progressive disease. And when the disease enters the secondary progressive state, you're in a neurodegenerative state that is irreversible. What you can prevent is those relapse and remitting phases, but by treatment, by aggressive treatment with drugs that target that immune response. I think similar mechanisms may operate in the context of PANDAS and PANS. If you treat the disease early, potentially you can reverse it, but I think there might be a point of non-reversal when the disease may enter in the chronic state that could be difficult. But again, this is my speculation. And I think more research obviously needs to be done on that.
I appreciate the window into your thoughts. The other question in relation to that is essentially whether your understanding is that individuals that develop PANDAS are effectively seeing repeated strep infections. They're interested in knowing how your team determined how many exposures to infection are sufficient to produce disease, and then whether there are any factors that were protective in mice that did not develop disease. I think you just touched on that a little bit, but if you could dig in especially to that piece of protective factors.
Yes, so in terms of the repeated infections, so in the study which is put in the bioarchive, but I didn't touch on it, what Charlotte did is a very thorough analysis of the T-cell infiltration into the brain after 1, 2, 3, 4, and 5 infections. And basically what she saw is that after 1 or 2 infections, you have very little T-cells in the brain. And where you have a massive infiltration is after 3 infections. And basically by the 5th infection, you basically have reached the plateau. So I think at least in mice, you need 4 to 5 infections to reach that plateau. And I think that the reason for that, we think, is that basically every infection triggers more activation of the immune cells, more maturation of Th17 lymphocytes, and then it also changes their phenotype to initially the Th17 cells produce only IL-17, and then they become what is called an autoreactive Th17 cells that generates IL-17 and GM-CSF. And these are the most pathogenic ones that are present in autoimmunity. And what Charlotte beautifully shows in the paper is that really this autoreactive phenotype can only be triggered after 4 and 5 infections. And so we really think that right now that you need multiple infections to trigger this autoreactive state. Unfortunately, children can get that in a school year. In a school year, they can get 3, 4, 5 infections easily. So even though we think that this is not possible for a child who is in school in 1 or 2 years of school, you could have like multiple infections. And some of these infections don't necessarily have to be infections that the parent is aware, sore throat, or because some of them don't fully develop, but they're sufficient to trigger an immune response. In particular, also we see this in mice. Initially, mice are very sick, but then after 3 or 4 infections, they kind of begin to develop a little bit resistance. Yet this predominant phenotype in the brain becomes very, very prominent after 4 or 5 infections. So there is a very interesting switch there. The other part of the question related to, sorry, I lost track here.
Oh, that's okay, Matt.
Was repeated in
It was a couple different pieces of the question. Let me go back to that.
Yeah.
The other question was what factors might be protective in mice that did not develop the disease?
I think the more important is what factors might be protective in humans. And
Well, I would agree with that.
that potentially, I think that we will be, in a year, probably will be able to tell you more. However, for those who are dying to know, my postdoc Urek Chan is giving a talk at the IOCDF Research Symposium in next week, actually, and he'll be talking about one of these.
San Francisco, that's right.
Yes. But yeah, that's one of the things that we've been very interested, not about the protective, but the more the triggering. So we've been interested in figuring out what potentially genetic risk factors could, trigger the disease or could predispose the children. And one of them is an important transcription regulator of cytokine secretion by microglia, which seems to be important in regulating the response to group A strep. Which is why I'm saying that, based on our genetic studies, what really seems critical is that, the innate response by the microglia plays also a critical role as well.
Great. And for those of you that are interested, that is the International OCD Foundation's annual conference in San Francisco. It starts on the 7th, it's in downtown San Francisco, goes through the 9th, and there are a number of presentations on PANDAS and PANS, as well as lots of presentations on treatment of general OCD. So thank you for that plug. Okay, there are a couple of questions here, thank you, about the olfactory bulb. So I'm going to do the second one, and the first is essentially about how the inflammatory response in the olfactory bulb might be related to basal ganglia, and then there was a little bit more in depth, so I'll give you this one as well. You showed that microglia exhibit a significant upregulation of CD68 in your model, as well as an upregulation of genes associated with, I apologize, I cannot pronounce this word, PHAGOCYTIC activity. Thank you.
Yes.
Do you think that T cell activation of these microglia as they enter the olfactory bulb are causing microglia to engulf the excitatory synapses that are absent in the strep model? In other words, do you think microglia are responsible for the absence of excitatory synapses because they engulf them in this inflammatory state?
That's a hypothesis. I think this is a very important, difficult question to answer when you target T cells. It will be a very good question to answer in the RXRA mutant mice. And in this transcription factor that is present in microglia, because one of its roles is also regulation of the phagocytic function of the microglia. And I think that that answer will be broadly elucidated in a few years. But I think that that's really what our sort of hypothesis is, that in fact, it is the microglia and their phagocytic state that potentially eliminates some of these synapses that are important for function. Similar to the elegant work done by Beth Stevens and colleague at Boston Children's Hospital, that she has demonstrated an important role for microglia and the complement in regulating these processes. But I think we're a little bit further from that. And I think because we really were lacking tools, and we also have some other tools that we can address the role of microglia in is a little bit more in detail, but those are things of the future.
Postdocs everywhere are paying attention right now to that question, I think.
Hopefully. Yeah, well, I think that question is very important in a large number of diseases like Alzheimer's.
Yeah.
And so I think that will become relevant.
We do have a number of questions about treatment and finding a professional. So I'll ask the wonderful folks in the background to post links to both the PANDAS networks. You can go to PANDAS Network and find a list of doctors there who tend to treat children with PANDAS and PANS. And you can also go to the PANDAS Physician Network's treatment provider list as well. And hopefully you'll find a good doctor through those lists. All right, I'm going to take a look through here the questions for just a second. Try to put a couple more of these together. All right. So there's a question here about that I just want to go back to. We'd asked about the value of antibiotics, NSAIDs, and steroids, and whether this confirmed it earlier, but we had missed the first part of that question, which was can this be measured in our patients in a way that currently confirms, excludes, or increases or decreases the likelihood of PANS? Is there a panel, for example, that doctors should currently be running? We have a couple different folks that have asked about those specific testing and whether there's a way to be helpful and then also to treat.
Yeah, so basically, I think there is something that, I can potentially, so basically among those cytokines and chemokines that I presented, what Charlotte did is that she ran an algorithm to predict whether she could see whether there was a good correlation between which of these could predict the patient and which could predict the control. And based on that dendrogram, there are several of them that were highly predictive of the patients. And so I think that not all 22 of them that were upregulated should be used, but I think there are a good number, 8 or 10, that are highly predictive that I think could be potentially beautiful biomarkers for the disease. And, some of the providers who are interested, I'm happy to talk to them more about that.
That's very generous of you. And this has been recorded and will be published in a couple of weeks and sent out via link to everyone that is registered. So if you did miss any of the list of the cytokines, that will be available to you via the presentation. And it is already up on YouTube from the last presentation that you so generously gave for the PANDAS Network's YouTube panel.
Yeah. I also want to point out, Susan, that we have also posted that in the bioarchive. So in the bioarchive manuscript, there is a table. The table that I presented is there. So, it's available. We made it available before the paper is out. The paper is currently under review. So under revision, actually. So, but we wanted to make it available to the community. So.
Okay. Terrific. We'll make sure that we post the link to that on the website as well. Okay, I have a couple more questions back to the olfactory bulb. This might be a little beyond your specific research on this, but asking whether that can cause olfactory hallucinations.
Maybe, but I will say that we certainly see a lot of defects in the olfactory circuit, and I think that it is likely that you could have a lot of like anosmia or like you can smell certain odors and you think that they're different odors, but I think that we haven't really dwelt into that much detail. But I want to come back to the question about the relevance to the basal ganglia, because I think it's an important one. And I think that what I want to point out is the reason we initially focused on the olfactory bulb is that basically this is the first relay center in the brain where a lot of T cells are coming. In our mouse model, the mouse model that we have, typically you don't get a lot of involvement of the basal ganglia, and that's just unique to this mouse model, but we are developing other mouse models. We're actually developing now a model that is called a humanized mouse model, where we are replacing the mouse antigen presenting genes with a human one, because Group A strep is a pathogen for humans. It's not a very strong pathogen for mice. And we think that we're missing parts of the disease by not really exposing them to the correct human antigens. And so stay tuned, because we are tweaking this model, and I think we're going to address some of the other questions. And of course, one possibility is to combine our model with a model developed by Madeline Cunningham, or the model that was developed by Ian Lipkin, to see if we can combine these models to generate a model that much better mirrors the human disease. One aspect that we are seeing is the involvement of the amygdala, which is a center that is important for fear. And I think that some of our future studies will also kind of focus a little bit on the amygdala as well.
On what's happening in relation to the amygdala.
Exactly.
Yeah, okay, that's fascinating. We look forward to all of your future papers. I think that we have covered the majority of the questions or combined them. I'm just kind of looking through here to make sure we haven't missed any. And so I'm just going to give you the opportunity to close. There's quite a few questions about how this might change treatment for specific children here today. And I wonder if you might like to speak to where we are in the disease, understanding of the disease as it relates to treatment today, and where we hope to be in the near future, next 5 years.
So I think that today, I think that there's a very, I think, through the Stanford and NIMH consortium, there's a lot of effort right now to really understand the disease and develop treatment protocols. And I think a lot of those treatment protocols, I would say, really strongly target what we have seen in the mouse model and what we're seeing with patients. So basically, they target the inflammation, as I said, that target the neuroinflammation. And then obviously some of the IVIG and plasmapheresis target at the, the antibodies that may be circulating in the patients. And together with some of the pharmacological treatments that target, like, more the neuropsychiatric components could be very helpful. However, as we know, there are patients who are still very resistant. And I think that is something that we really need to pay attention, because I think a lot of the clinicians and scientists as well, we love nice stories that fit, but then we have to also think about those cases that don't fit. They don't fit like the easy picture, right? And sometimes the cases then can come to the clinic or to the research questions are those patients that haven't really gotten a good treatment from the get-go. And I think that really what I described today is more, something that really think in the long term, like, are there cases where we really need to consider therapies that are being already developed in other diseases that we can repurpose for PANDAS and PANS. But also broadly speaking, when we think about obsessive-compulsive disorders, right, there is a subset that is clearly triggered by, has an immune base. And I think even when we consider like adolescents or adults with OCD, and whether a subset of them could be like PANS or PANDAS that develop in adult OCD, I think it's important because some of these therapies could be beneficial to them as well, particularly immune-mediated therapies that currently are not considered when you're thinking about OCD. What to me was also very interesting is that just the picture that we see with microglial activation is very similar to what you also see with how microglia get activated in either neurological, psychiatric diseases. And so to me, that also brings PANDAS and PANS central to this concept that it is a neuroinflammatory disease. And I think if, clinicians ignore this right now, I think would be detrimental for the patients, because I personally was shocked to see how similar those cytokines were between the mouse and the human, because we had gathered the data from the human, and we really hadn't published it because we were trying to make sense of where they were coming from. And we really didn't make a lot of sense until we actually did the studies in the mouse and we saw which cells were making them. And to identify that really the majority of them came from microglia and macrophages, really to me suggests that indeed these cells, like the activation of the innate immune response through potentially T cells is central to a lot of autoinflammatory diseases, and makes a lot of sense for us now to think that PANDAS and PANS isn't the disease, and therefore should be treated similar, I think, to what we think about MS. That's my point of view.
I appreciate your sharing that. I think in addition to the really dedicated work that you and all of your collaborators are doing, I often speak of your empathy for the patient, especially the difficult patient, the patient that is treatment resistant to some of the current therapies that we have. That's deeply appreciated. And then also your willingness to collaborate and share information, both with doctors who are trying to treat as soon as you know information, and also with other researchers that are looking at this through different lenses and trying to understand how we might develop good treatment therapies and diagnoses in the future. So thank you for that from all of us.
And I think, Susan, what I want to also emphasize is our study is very small, right, in terms of the human study. So I think for me, it would be really important. It is very important, and I will take no offense if basically other studies can reproduce all the cytokines. But at the end, the power of the disease comes from the numbers, right? The power of the genetic studies, the power of the cytokine profiling comes from the numbers. So I really urge all the clinicians who have cases of patients, we are very happy to collaborate, or you can run the cytokine panel yourself. But I think it's important to really kind of see how our data integrate with other data, because the validity of the study will strengthen, of course, by reproducibility, whether it's an independent or, but I think it's also important to look really carefully at the cohort. I think one of the most difficult things is really to know like where your patient is. We made sure in our study to really have cases that were acute, because we really didn't want to confound an acute and a chronic case. And I think it's really important that a clinician really looks at this very carefully. The clinical history of the case, like when was the patient diagnosed? And I think how that, because a chronic form of the disease will represent very differently from the acute, even in the cytokine panels that you see in the blood and the CSF. And so I think that it's important to really be careful about this. And some of those patients who are refractory are likely to be chronic. So I think it will be important to characterize those carefully. Mm-hmm. And to me also would be very important to see where do we see how much convergence and divergence is in the disease depending on what pathogen triggers this, right?
Right.
Because, we could be in a situation where all roads lead to Rome, but the causes of how you get to Rome are very different, or the roads. And so at the end, the manifestation of the disease could be similar, but the mechanisms could be different, which means the treatment would be very different as well.
Right, especially for the primary infection. Right. It's really quite fascinating, because when you look at a group of patients and try to think about this disease, you have to ask the question of whether they're in the acute or the chronic phase, and you have to ask the question of what was the pathogen or the trigger that started the disease, and whether you're going to get different answers on that. So it certainly makes your work very complicated.
It does.
Ask away. All right, wonderful. Well, with that, thank you. We have appreciated your generous time here today. I also want to thank the PANDAS Network for all of their generosity in fundraising for research in the field, and also for their generosity in sharing all of that information with both parents and treatment providers. And of course, to the PANDAS Physician Network as well for the dissemination of information and research into PANDAS and PANS. We certainly hope to change both diagnostic and treatment hope for this disease very soon in the near future. So thank you again for being with us here tonight. It's much appreciated.
It's a pleasure, and thank you for hosting me. I appreciate it.
Anytime.